Hi all, this is Dr. Prasita, a co-author of this book. So I would like to explain a few things on the applications of nanomaterials through the green synthesized ones. Okay, so it's like nano-sized materials are popularly synthesized by various physical and chemical methods which are not environmentally friendly. Then what could be the best way of synthesis of nanomaterials, you believe? It is biosynthesis, or it could be also called green synthesis. Why? Because the biosynthesis of nanomaterials is a green approach as it is safe and eco-friendly. To add on, it's prompt and even cost effective. The required plant part is cut into pieces and crushed using mortar and pestle to obtain the plant extract. The extract is simply mixed with a solution of metal ions at room temperatures. The reaction is complete within minutes. Many nanoparticles have been produced this way. These biosynthesized nanoparticles or bio-nanomaterials have been used in a variety of applications, including drug carriers for targeted delivery, biosensors, bioimaging, tissue engineering, coding, protein detection, cancer treatment, gene therapy, and DNA analysis, or antibacterial effect, and even in wastewater management. As we see here, these biosynthesis nanoparticles or bio-nanomaterials have antibacterial activity and possess many extraordinary properties, due to which these nanoparticles are very useful for biological applications. So what is an antibacterial activity? So antibacteria means against bacteria. So the antibacterial activity of a molecule is that it kills the bacteria or gradually diminishes the rate of growth without being extensively toxic to nearby tissues. Usually, antibiotics act against bacteria which kill or prevent them from reproducing or spreading. Okay, so here we have our bio nanoparticles which acts as an antibiotic having antimicrobial activity that has been demonstrated broad spectrum antibacterial properties against both gram positive as well as gram negative bacteria. So antimicrobial activity of nanomaterials can be studied using three main methods: disc diffusion, broth microdilution, or agar well diffusion, which are standard methods recommended by Clinical Laboratory Standards Institute CLSI for measuring in vitro susceptibility of bacteria to antimicrobial agents. Among these, agar well diffusion method is widely used to evaluate the antimicrobial activity of these bio-nanomaterials. The disc diffusion test is also known as the agar diffusion test, Kirby-Bayer test, disc diffusion antibiotic susceptibility test, disc diffusion antibiotic sensitivity test, or KB test. Now let's see what happens in the agar well diffusion methods. So we normally use petri dish or petri plates or agar plates. So the detailed description can be seen in page number 195 of this edition, that is the section 3.2. The detailed procedure of agar well diffusion method has been defined. So we can simultaneously test large number of antimicrobials in an easy and flexible way. So Mueller-Hinton agar broth, that is MHAB, is usually the nutrient agar medium used, which contains beef extracts, peptone, sodium chloride, and yeast. So it is sterilized at 120 degrees for 15 minutes and poured to sterile petri plates so that it solidifies in an airflow chamber. After solidification, with a cotton that is sterile cotton swab, the bacterial broth culture is swabbed on agar plates and the wells are made by well cutters. Then the samples are loaded into each well and the agar plates are incubated at 37 degrees centigrade for 24 hours. After incubation, the plates are absorbed for zone of inhibition around the well, and the diameter is measured by a ruler or just as your normal scales. So based on the diameter of inhibition zones, the results can be assigned to three categories. That is, the bacteria can be either susceptible, intermediate, or resistant to the antibiotic used. So the bigger diameter of the inhibition zones indicates that the higher antibacterial efficiency is present for the samples. So the detailed description of such things can be seen clearly here. Hope it's clear. Thank you.